All right, thank you.
And please go ahead. Thanks.
And thanks for sticking with us till the end of a long but very exciting conference day. I will try to keep it still entertaining and to keep your alertness awake, and myself. Also trying to connect to some issues that we've discussed today, especially to the previous talk about the microbiome and its interaction with the hosts through the immune system. That will be a major component as well. And also tying back to something that Jonathan mentioned in his talk about widening the scope of what immunological recognition is all about. So that's the reason for the subtitle, that maybe we are moving towards a new theory of immunological recognition. And in general, there seems to be a kind of transition in theoretical conception as immunology moving away from the paradigm of it being mostly something that fights pathogens, which of course now is on everybody's mind, to fight certain viruses as effectively as possible and also to develop vaccines. But along the same lines, the attempts to develop vaccines compared to the success stories we did have in our history of modern medicine and the other diseases where vaccination attempts are still struggling to find effective means for the immune system to be able to recognize and then respond to certain entities and activities in the appropriate way that we want them to, might require some rethinking about the very first component of how the immune system recognizes the objects it deals with, or even the processes. With immunological recognition, it used to be the case that, well, we've all read the textbooks, and in Charles Janeway's influential paper in 1989, in which he was really opening a new perspective about how the innate and adaptive immune system interact. But on the status of immunological recognition, he was rather suggesting that we were approaching an asymptote of knowledge, where we can still, in future experiments, figure out the more technical details, the higher degrees of precision, the digits behind the comma, rather than real revolutionary changes in our understanding of how immunological recognition works. Well, probably, as also agreed in most of the immunologists today on the research front, that there might be needed a conceptual update to the rather molecular reductionist conception of how immunological recognition works. And this will be what I will try to paint in a big picture view very quickly. The outline will be to give a very, very brief history, so just one slide, very few keywords of the history of how this now standard theory of immunological recognition came to be, and then argue that it's much wider than just the molecular patterns binding onto each other like lock and key on the structural level, that there are indeed other notions of structure that play an important role in immunological recognition as well. And in addition, maybe the most innovative part will be to argue that the immune system will actually recognize some sort of functions in addition to structures. So we all probably heard about and seen in particular this famous picture of Paul Ehrlich's side chain theory, when immunologists were still fighting whether immunology was rather about the body fluids or the cells, where Paul Ehrlich proposed that the antibodies were rather due to their form fitting like Legos onto certain receptors of cells, which also fitted nicely with the popular metaphor of a lock and key, coined by Emil Fischer. Later, it was thought that immunological recognition was all about distinguishing self from non-self based on McFarland-Burnett's theories from the late 1960s, which were for a long time really successful and were updated from the distinction to just have self and non-self for obvious reasons that sometimes immune systems also work against self components and tolerate non-self ones, that Polly Matzinger updated her theory that there was rather a distinction made between dangerous and non-dangerous components. But still, everything was kind of moving in this, what people have been calling the chain paradigm, that would be a corresponding between one molecule binding one receptor that would lead to one functional response by the immune system. And that is about to need to get a conceptual update. And here's a philosophical attempt to give that. Structure is a polysemic terms, and especially in immunology, what people have in mind first and foremost is the three-dimensional molecular shapes, like of this beautiful drawing of an antibody right here by David Goodsell. And they will be binding with high affinities to the according receptors, all in line with this one molecule, one receptor, one response chainway paradigm, which has been extremely successful. But also there are the limitations to this framework. Another important discovery, especially when it came to proto-immune systems in microbes, like the CRISPR-Cas system, which doesn't have such a sophisticated machinery or repertoire of different binding molecules, but here it's rather about the sequences of foreign DNA. But as Thomas Bordeaux and Jean-François Moreau have argued, maybe it's also not so much the property of being foreign, but rather that some genetic anomalies are the important factor whether or not a proto-immune system in bacteria will react to such DNA sequences, which also makes more sense than how would it know what is self and what isn't. It doesn't have a real comparing to a library, but rather searching for genetic anomalies on a more functional level, perhaps, is probably a more reasonable way to think about these things. Anyway, so we also need to have sequences, so structure, primary structure of sequence molecules in mind. Then more complicated, we have mathematical network structures, which here are still very simple, but can become large, complex networks quite easily. Again, here these are very simplified mathematical toy models, but already these toy models can be useful to explain some very complex and interesting dynamical features of such components. And of course, if you have not just 3 nodes, but a multitude of a complex network nodes interacting, the picture becomes much more interesting as well and needs to be analyzed with mathematical tools. Also links to the dynamical outputs that also Jonathan mentioned earlier by the discontinuity theory. I'll also come back to that later, because these are properties that can't be really attributed to just one molecule binding one receptor. There's more going on. These kind of structures also need to be taken into account when it comes to immunological recognition. With the microbiota, there is a ton of things that these do. One of is which is providing certain metabolite precursors or other molecules that acetylate and interact with the host. And they have, of course, 3-dimensional structures. But again, we are not believing anymore that there were like a kind of barcode, that these molecular 3-dimensional structures were like labels or flags or barcodes that would allow us to identify, oh, this is this particular type of microbe, and therefore the immune system will respond in this particular way. For example, short-chain fatty acids is one metabolite that is produced by a diversity of different bacterial species. And the immune system still is being interested, to use this theological metaphor, that these are being produced and doesn't really care by whom, or the response doesn't need to be taking into account by whom certain molecules are being produced, rather that they are being produced at all. And the immune system in its interaction with the microbes, most of the time is not a response of fighting them as pathogens, but rather tolerance is the rule, especially in the gut microbiome, where we have this the more enormous cell count and genetic diversity, where the immune system is not only not attacking them as pathogens, but really actively contributing and shaping its sometimes that it will be according with the physiological maintenance of the whole organism. And so the immune system, again, is not just there to fight pathogens, but is involved in much more processes like repair, maintenance, physiological and metabolic processes, growth and development, and so on. In a nutshell, we could probably sum this up that maybe it's not so important who is there, but rather what is going on. So when microbial studies look at the taxonomic composition, mainly of these microbial samples, they might miss the more interesting component of what is going on. But granted, this is much more difficult to do. Even if you sequence the whole genomic sequences are in these samples and try to link them to functional features, there's a lot of problems doing that. And at least it's a good first step to get the inventory of who is there. But at least I will argue that we cannot stop there. And for the real understanding of how they interact with the immune system, with the host organism, we should be rather focusing on what is going on. In addition to the zoo of what in immunology is called the pathogen-associated molecular patterns or danger-associated molecular patterns, I will then suggest that maybe we should introduce a new class of molecules that I will call function-associated molecular patterns that are still molecular products, three-dimensional molecules, but they are indicative of some functional aspects like producing certain vitamin precursors. So this is not associated with a danger or anything pathogenic, but rather something useful for the host organism. And the immune system will still recognize that and respond to that, not in this way of like fighting the microbes, but in the opposite, maintaining these kind of microbes to keep doing what they are doing. Or at least tolerate them and leave them alone. So to sum up, the immune system can recognize different kinds of structures from the three-dimensional molecular shapes to network structures, also including those produced by microbes. But how about functions? That might sound at first like a crazy idea. How could the immune system recognize functions? Well, if we want to still go with the molecular associated patterns, I already suggested that maybe we should broaden our perspective in classifying them into pathogenic, microbe associated or dangerous, and also be open to have function-associated molecular patterns that are indicative of useful contributions by microbes and not by dangerous and pathogenic ones. Even in a recent immunology textbook that is named after Charles Janeway, of whom we are trying to give a conceptual update, it is being acknowledged that the recognition part of these associated molecular patterns has become aware that there might be an increasing number of other receptors that are capable of discriminating a number of closely related molecules. So there might be much more greater diversity of these receptors and the way recognition works than has been previously thought. So against the view that we would have reached an asymptote of knowledge and just need to figure out some last details. Actually, if we take this functional perspective, there might be a whole new, opening up a new research perspective of what other kind of function and activity associated patterns the immune system might be monitoring. So I think I will speed up a bit to keep the time, because most of this we have already just discussed. Here is a figure where I want to illustrate this point. So far, the view of immunology and its recognition has mostly been focusing on structures and classifying them into being associated with certain microbes or being with certain danger classes. We can open this up to a more neutral and useful perspective as well, where still molecular structures will be indicative of certain functions, roles, and effects they have physiologically on the organism of their hosts. But also there is other functions that are not associated to certain microbial, certain molecular patterns. So think of things that take place on a higher level to maintain a certain provision of vitamin precursors over time. It's not something that's directly linked to this molecular pattern. Or there is things like heat and pressure, the things going on larger scales, and maybe a more convincing concrete example I will discuss in a minute or maybe just to say the general before that there is many enzymes that do the same things and have different structures. And even if they have the same biochemical activity, the effects of these molecules, the functions on the higher levels, can be quite diverse, whereas the molecular patterns, it cannot be linked to any concrete molecular patterns, as in the case of the conventional picture that is very structure focused. So what is going on with these functions here? Matzinger's danger theory still tried to classify the structural space according to these self, non-self pictures, or dangerous, harmless, or infectious pictures, which in a way is useful. You can do that in the structural space, and it helps a lot, but at the neglect of the functional space, where many undiscovered targets of immunological recognition might just sit there to be discovered. But how should we talk about this dashed cloud of functions? And philosophers have been talking about function quite a lot. This debate is always between these two classical accounts, but I do think that the selected effect picture is out of the game, because we would hardly want to argue that the immune system recognizes why something has been selected to be there. That would be a very weird story to tell. But rather what the things are doing causally, mechanistically on this level. But here we can attribute biochemical activities to single molecules, but then what role they play in a larger mechanism, that's also something that you cannot always attribute to an individual molecule, but you would need to attribute rather to a network of interacting agents. The need for a defining function is also acknowledged by microbial researchers. So more interaction between philosophers and scientists is clearly needed here. Also when it comes to defining the structure and function of the microbiome. But I think for reasons of time, I'll just really rush through this with the same story, that maybe what is going on is more important than who is there to understand the interesting questions. So more emphasis should be put on the functional aspects. Because the microbiome also can be decomposed with different notions of structure and function. And many people have been arguing, mostly from an evolutionary perspective, that the functional aspects might be more important when you compare the entities, the players. So the individual microbes might change, but still they are having similar effects. So there is very witty words, phrases for that, and brilliant papers. But on the physiological level, how could it be that the immune system recognizes these kind of functions? Well, when it comes to antibody binding, that's a biochemical activity that can still be mapped onto an individual molecule, but whether or not what kind of immune response it triggers, that's something on a higher level, what biological functional role this plays in the organism. This is, of course, highly relevant also for vaccination attempts, because most of the vaccines are reversely designed that there will be binding of certain structures, antigens, which of course, that nicely works on the biochemical activity level of individual molecules and their binding activities, but whether or not it will be triggering an appropriate immune response, that's something that takes place at the level of functional roles and interaction networks. And this is why a lot of the struggle at the late stages of immune trials, vaccination trials, can fail, even though the first promising results about the binding were very promising, but then whether or not an actual appropriate immune response is being triggered without side effects, that's much more tricky and on a different level. So here's a very quick and I think convincing example of a functional entity, an enzyme in Drosophila. That's an immune sensor for proteases. And you can see there is dozens, if not hundreds, of different pathogens that have different molecular structures of these proteases. And the immune sensor is not detecting any of these individual structures, but rather the effect of the biochemical activity, that is the protease activity. So any of these structures will cut a bait region of this sensor, and this effect will be the signal for the immune system. So the immune system recognizes not any individual molecular structure, but here is protease activity going on. It's something functional and not structural. Immunologists have also made a similar case in the past, so it's not just a philosopher's crazy idea. But here they have been mostly arguing that the recognition of functional features should be something to take into account when it comes to multicellular and complex pathogenic entities, whereas the microbial in the microbial domain, things would still be dominated by structural feature recognition. Well, if we generalize the account, I think a good convincing argument can be made that also functional feature recognition takes place at the level of the microbial entities. But because I think the reason why scientists were more open for this kind of thinking on these levels is because here many attempts to link immunological recognition to any molecular shapes have been failing and so been proven so difficult. Whereas on the microbial level, molecular patterns approach has been quite successful, but it also has its limitations. So I think the idea that the immune system is recognizing not just structures, but also functions is not crazy, but actually very useful in progressing our understanding of what the immune system recognizes. There would be many ideas to speculate about and look for more concrete examples. When I talked about pressure, that was actually something that was just a speculative idea, but just a few months later in a nice Nature paper, it was actually discovered that there is an immune sensor for pressure, which they call pH1. I can give you the reference if you're interested in that. So just wrapping up, in addition to individual different notions of structures, the immune system also can recognize different kinds of functions, including biochemical activities and the biological roles of these activities in larger mechanisms. Taken together, it will also be the case that these different kinds of structures and functions will also be recognized by the immune system, not just for being there in absolute terms, but the spatio-temporal patterns of that. And I think here it's very interesting to see that the presence of antigens, as they framed it in this paper, will give rise to different immune responses depending on the spatio-temporal pattern in which this antigen is present. But you can easily substitute the antigen for biochemical activities and biological roles. And I think this will even further help understanding what's going on sometimes with the immune system, recognizing or not recognizing certain molecular patterns, structural features, or functional features. All right, time to summarize. So I think that the traditional picture of viewing immunological recognition based on three-dimensional molecular shapes is too limited. And instead, we should open the possibility that the immune system can recognize functions as well, where we can go into a deep analysis concept of philosophical notions of structure and function. I gave you a very quick overview, but I think that both the introduction of a function-associated molecular pattern is a useful one, and even more so opening up the space of functional features that are not linked to any particular molecular patterns. These claims are not just armchair philosophy, but I think there are good case studies in science as well which support the claim. And again, the wrap-up slogan is for immune system, especially in interacting with the microbiome, that what is going on might be the more important component compared to who is there. And with that, I will thanking you for listening and staying up with us.
Thank you. Yep, let's thank you virtually as well. And Jonathan.
All right, so can you hear me all right? Good. All right, thanks a lot, Gregor. It's a really interesting talk. And one thing I've been interested in, I guess, is I've been reading more about these issues within microbiology and this kind of shift towards the need for more focus on functions rather than phylogeny. I mean, partly I wonder to what degree this is going to complicate matters quite a bit, in the sense that, as you said, I guess these functions are going to be perhaps now attributable to a systemic level or a network level, and perhaps they're multiply realizable. So, I mean, in what sense do you think we would be able to kind of overcome some of these challenges in establishing kind of causal explanations when you shift from kind of structures which seem to be somewhat easier in a sense because they're somewhat more discrete to functions.
Yeah, that would be a good, brilliant question also, because it raises a lot of the methodological consequences and concerns when, of course, it makes sense with the easy approach and reductionist picture first, and the tools for analyzing networks, bioinformatical tools you would need to really dive deep into this systems level analysis. There is a lot of work to be done, both conceptually, but I think it will be helpful mostly in avoiding confusions and wrong expectations and treating things as a kind of black box and then failing to have any idea where to look for the actual explanation when exactly for the question where do we attribute the health to on this level, by just thinking it is a property of a particular taxonomic composition would fail to recognize that there is much more going on than just having this an ensemble, and mostly avoiding confusion will be the philosophical contribution, and then the scientific challenge based on that, if it's any sound, what we argue here as philosophers, would then be exactly to find the right tools for tackling exactly these questions.
Okay, great, thanks.
So, time for additional questions. Let me ask, I have a couple of them. So actually, this is just a sort of a, let's say, terminological question. So you were referring to them as danger-associated molecular patterns. I've always thought that it stands for damage-associated molecular patterns. Is this interchangeable, or?
In the literature, you can find both. I mean, Polly Matzinger, she was all about her danger theory.
Yeah.
But it's true, like maybe more common in the literature, people say use damps with danger. That's true, with damage. Yeah.
Okay, great. And then the other, I guess for that, it would be best if you could put your slides again. Mm-hmm. So there was a slide with a quote about vaccination.
Yeah, by Van Regenmotel.
Yeah.
Here.
Yes. So could you just quickly go through this again? What does the quote illustrate? What?
I think it's mostly Mark van Regenmotel has been arguing against the reductionism, especially when it comes to vaccination, because it confuses the levels at which an organism can be vaccinated by just focusing on, oh, we need just an antigen binding to an antibody, and once we have that, it will give us immunity. Well, that's clearly a too shortcut picture, because of course you can have perfect binding without triggering the immune response. Often actually it's the opposite, that it needs a too tight binding will stop the machinery, and just a certain amount of binding and the what's the verb I'm looking for, like the dynamics, the binding dynamics of on and off states will be more important than binding too tight, especially for CRISPR-Cas system. I think also a perfectly fitting target sequence will actually stop the machinery. But if you have a binding dynamics of off and on states at the right level, that will be actually the most optimal way for that. So in order to vaccinate an organism, you can't neglect the organism level and just say, oh, I have my antigen binding body, job done.
Yeah, so I mean, basically, it seems to me that to say that you can't vaccinate tissues or organs, rather, in some sense, I guess you can, because there is a debate about, for instance, the route of delivery. So if you vaccinate intramuscularly as opposed to other ways of routes of delivery. So for instance, you can get a boost in IgG production, but not really IgA production, and this can have major implications. I mean, this is also the case with COVID, for instance. So you can get a good response and production of IgG, so that you can get protection for a lower respiratory tract, for instance. but not really when it comes to the upper respiratory tract, where IgA is much more effective and substantial. So then, in a sense, I guess you could argue that there is a way, like this reductionist way of thinking, that you can vaccinate specific organs, like you know, the mucosal surfaces, as opposed to the organism as a whole.
Mm. Yeah, I mean, sure, the site of vaccine administration is really important, but I think I would refer to Jonathan's brilliant answer on my question earlier, that even though maybe you could say that also to this extent the organ has a kind of vaccination protection required, that, but the actual unit at which you want to say that some entity has been successfully vaccinated still is the organism.
Yeah, I guess that's about right. So that was all from me. I don't see any hands raised, but please don't be shy. We are roughly about to hit the time. So if no one raises his or her hand, then I guess it's time to conclude. So thank you, Gregor, again.
Thank you.